how much progress have you made in vaccine development?
Well, we fell so many times. I don't think, if I told you all our experience and our work in African swine fever vaccine, is going to be very long for our people. So I'm going to reduce a little. We work very much in Spain, not only my group, many other groups as well, in vaccine productions, because was a disease that is very complicated to obtain a vaccine. One of the reasons why that's happened is because it's probably the only virus that affected animals that don't induce neutralizing antibodies. So the neutralizing antibodies for our friends is the antibodies that really kill the virus, not the ones that react with the virus, but they also kill the virus, they immobilize the virus. So these antibodies are not produced in ASF, or at least we don't find the protein that can induce these antibodies, or other proteins are blocking to that protein, so the antibodies are not presented to the immune system, the antigen is not presented, and is not neutralizing. So it's a complicated virus for a vaccine. We found that the inactivated vaccine doesn't work. We found that some deleted vaccines, vaccine that you take genetic, is working. Some of them are working, but not well. So the vaccine, all the trials that until now has been used, prove that the best protection is obtained with an attenuated vaccine. So in 2017, so 2 years ago, we was very lucky, and we found in Latvia, our colleagues from Latvia sent it to Madrid. They sent us some sample from one wild boar that has been shooting in Latvia, and they sent it to the European Laboratory Center in Thessaloniki in Madrid to see if it's died or not, or if this animal was carrier or not of African swine fever. And we isolate one virus that when we look by genetic and we study the sequence, we found that there are changes in 2 genes that we know are very important for the virulence and for the protection. So we thought this attenuated is going to be attenuated for sure, so the animal doesn't die for African swine fever, so it's an attenuated one. So my colleagues in Madrid, the first study was in domestic, so my colleagues of the European labs, they did the first inoculations in domestic pig, and a very few ones, and they found protection. So they told me, because I tell, please put that in the pigs. You have 5 pigs, see what happened. So they told me, and I said, okay, let me the virus, and we're going to work festival and wild boar. You're probably asking why wild boar? Because living in Europe, and in 2017, wild boar is our best threat.
Right.
I mean, the most important one. So we dedicated a lot of research to see how the virus work in protected wild boar. So we found that it's orally was beautiful, they immunized beautiful, and the protection was very good. The protection was 92.75 of protection. So not only was well protected, also the animals that has been vaccinated and challenged with Armenia. Armenia is a very high acute virus of the area, of this area. So the animals not only survive, but they also have no lesion at all. And also the virus of Armenia was not present in any lymph node. So the protection was clean, what's what we call a sterile protection. So we don't find any piece of virus in the lymph nodes, not from the attenuated vaccine, not from the virus one. So these results really, was very, 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 very stimulating us. And so we really think that we have a good prototype of vaccine. We are now working in the security of the vaccine, because one of the problems that the attenuated vaccine could have is the reversions to virulence. So that means that you have a virus that is attenuated, but after sent it or put in different animals, they reverse to the original virulence. So we have to prove that these mutants are stable. So this is one of the work we are doing, working with other resistant and also with a lot of testing for security. So if we pass the security, if all the tests that we are doing pass the security, I think we're going to have a good vaccine, a very good one.
The last thing you want to say to Korean farmers and Korean government?
Well, I would like to, first of all, to congratulate you for this wonderful country. I think it's very good, and I'm really very impressed with this country. And so my first words is congratulations for your country and for your people. And the second one is you are in risk to be infected with African swine fever, and please do as much as you can. So I know it's not easy, but at least we know which are the risks or the main risk to be introduced to your country, so you can probably stop most of them. So the most important for the administration is tell the people about this risk, tell the people how distinguish this disease of other, and stop the potential entrance of the wild boar, the potential entrance of swill feeding, etc., etc. And to the farmer, please be our eyes. The farmer is our eyes in the field. If you detected any similar symptoms, the ones that we already tell you, please call your authorities. As soon as we stop the disease, it's going to be easier to clean everything in this country. And this country is wonderful, and you too. So do it. You can do it. Thank you.
그리고 돼지병터에서는 앞으로도 아프리카 돼지열병이든 다양한 양돈 업계 소식에 대해서 혹은 양돈인에 대해서 인터뷰를 하거나 하면 계속해서 지속적으로 업데이트를 하도록 하겠습니다. 감사합니다. Thank you.
Thank you.